Biotechnological applications in medicine. Recombinant DNA technology. Recombinant DNA technology involves the manipulation of the genes or microorganisms to produce certain products which are useful to the mankind. That earlier we identified that particular microbes are useful for the human welfare. Now, because of the recombinant DNA technology, now we came that we are altering that particular bacteria to be useful for us by introducing the genes, certain required genes into that particular microbes, and we are making them for the products of human products we are producing here that we call recombinant DNA technology. And these recombinant DNA technological process have made an immense impact on the area of human health care. They are used in the production of hormones, that synthetic vaccines. Recombinant DNA technology is making facilitating the production of hormones, synthetic vaccine, effective therapeutic products. Likewise, so many products we are producing. These products like the recombinant therapeutics do not induce unwanted immunological responses. They do not induce any unwanted immunological responses. At present, 30 recombinant therapeutics have been approved. 30 recombinant therapeutics are approved for the human use the world over total. Okay, so genetically engineered insulin is one of the best example to say about the production, how the recombinant DNA technology is getting used for us is the bigger one. Diabetes mellitus is the biggest problem. The worldwide diabetes mellitus, in those people, the insulin is not sufficient or insulin going to be absent. I'm talking type 1 and type 2. In case of type 1 people, diabetes insulin is no more. By birth, it may come even. In case of type 2, that late onset diabetes we discussed earlier, so in those insulin, the levels are going to be less. So that means what is the treatment for those people? Insulin injection. Okay, insulin is a protein hormone produced from the beta cells of the islets of Langerhans of the pancreas, that endocrine portion of the pancreas. We used to say islets of Langerhans, and that having beta cells that producing this insulin, which is required to accelerate the uptake of the glucose. We know the function of the insulin, the conversion of the glucose into glycogen done by liver cell. What is the role of insulin then? Insulin that increases the cell permeability. That means that intake of glucose by the liver cell. Insulin facilitates the permeability of the glucose into the liver cell and make it conversion of the glucose into glycogen. This is the basic function we discussed in chemical control and coordination, endocrinology chapter. Okay, so that in those people which the insulin is not sufficient, so what is the requirement to them? That insulin is to be injected over, must be injected or given via an insulin pump. Okay, insulin for glycogenesis, insulin for lipogenesis, insulin for protein synthesis, very, very important hormone. In those people who don't have insulin or who are having less insulin, they suffered with a disease called diabetes mellitus. For them, it is must to be injected insulin or given through the insulin pump. It cannot be administered orally. It cannot be administered. Insulin you cannot take through mouth, through the as like a tablet form. Why? Why here? We know that in the stomach, gastric juices are present, pepsin is present. So, and this insulin is a protein hormone. When you take insulin, what happens? That automatically that particular insulin will be digested over, isn't it? So that's why you cannot take administered, you cannot take it orally. So that's why injection only the form here. But still, the efforts are going on. Attempts are underway to develop orally administrable insulin. But at present, still the diabetic patients are depending on insulin injections only. Okay. The problem I think you might have understand that why we are not taking insulin through oral. That means when it taken that itself in the stomach, that insulin may break down digested form because of the enzyme pepsin. So prior to the development of the humulin nothing but insulin. Human insulin we call humulin in general. Diabetic, before we produce the insulin of a human being in recombinant DNA technology, the diabetic patients are used to be dependent upon animal insulin extracted from the pancreatic islet tissue of the slatted cattle and pigs. From there, we use it to collect. Okay, that insulin is collected from the that pancreatic islets of Langerhans of slatted cattle and pigs. The animal insulin is more expensive. That previously when we whatever we used is less accessible and causes allergies. Why? Because that is foreign body. We that insulin you can take, but along with the insulin, when it is extracted, the other proteins of the animal proteins are also can be seen over. So we consider them as antigens, and what you produce, antibodies, several side effects will be seen over. We call it as a hypersensitivity, serum sickness, particularly in them. So currently, genetically engineered, that was the problems we faced earlier. That currently, genetically engineered Escherichia coli, which can be grown in large quantities to produce the human insulin, which is called humulin, is available and from which we can make much insulin as we need. That means whatever the insulin, we call it as humulin, now available in the market, that is produced in the recombinant DNA technology. Okay, so then let's talk about that structure of insulin. To understand how insulin is synthesized, we need to know the structure of insulin. That human insulin, as we already said, it is a protein hormone made up of 51 amino acid. 51 amino acid arranged in two polypeptide chain. So we have two polypeptide chains. One is A chain, A polypeptide chain, which I am showing here. This is the A polypeptide chain. And another is the B polypeptide chain. A polypeptide and B polypeptide. So A chain contain 21 amino acids are present in the A chain, and B polypeptide chain contain 30. Total number of amino acids present in the insulin are 51. 51 amino acids are present, which are actually connected by disulfide linkages. You see here, yes, that is yes means disulfide. So finally, the structure A chain and B chain. A chain containing 21 amino acid, B chain containing 30 amino acid are connected each other by disulfide linkages, which is called as insulin as a protein hormone. Insulin as a protein hormone. So that's it. That is about the structure of insulin. Okay, in mammals, including human being, when we synthesize the insulin, we don't directly produce the insulin. That means we are talking about the beta cells in the islets of Langerhans in a human being, in any human being. Okay, insulin directly is not produced. Insulin is as a pro-hormone, it is produced over when it is produced from the beta cell. Like you might recall pepsinogen, trypsinogen, chymotrypsinogen. These words are familiar for us in digestion absorption. What we used to say in them, pepsinogen is an inactive enzyme. So what we have to do, we have to make them active. We call pepsinogen, trypsinogen are pro-enzymes, isn't it? So these are pro-enzyme. Conversion of the pepsinogen into pepsin require HCl. Conversion of the trypsinogen proenzyme trypsinogen into trypsin by enterokinase of the intestinal juice. So that we said earlier. Similarly, in the human insulin, when it is synthesized, it will be synthesized in the form of a prohormone. So that's what we said, like a proenzyme which needs to be processed before it becomes fully mature and functional hormone. Okay, so that prohormone you can see in the diagram. This is the prohormone. This is the prohormone which I am showing in the box. Okay, which contain extra stretch called C peptide, which is shown in the gray color. I'm using black. Here it is. That is C peptide. Okay, so the A, B, C, if they are present, what do you say? That is the prohormone. So this C peptide is not present in the mature insulin and is removed during maturation into insulin. Likewise, what we said in case of pepsinogen to be converted into pepsin, the beta cells, when they produce the insulin hormone, they produce in the form of proinsulin, which contain A polypeptide, B polypeptide, and also containing C polypeptide. That C is there in between. The C peptide is present in between, and maturation means like this. the C peptide will be removed, and the free C peptide, you see here, you see the free C peptide will be removed, and then insulin will come under function. So that's it. That is the primary information we need to know. What kind of problems we faced over during the production of the recombinant recombinant DNA technology when we are producing this. What kind of problems we faced here? To know that, this is a very important information. Okay. The main challenge for the production of insulin in the laboratory using rDNA technology, recombinant DNA technology, was getting insulin assembled into its mature form. When you prepare A, B, C, how to make that particular insulin to be matured form, that is going to be a tougher task. In 1983, L. Lilly, the American company, LLE, American company, prepared two DNA sequences. They got an idea. They prepared two DNA sequences, one code for A and one code for B. Separately, they prepared human insulin. A containing 21, B containing 30 polypeptide chains are separately prepared and introduced that two genes are separately they synthesized, artificial gene, we can say. DNA sequences, one DNA sequence for A and one DNA sequence for B, separately introduced into the Escherichia coli to produce the insulin chains, and the chains A and B were produced separately. The two DNA sequences they are taken. In our language, we can say two genes are produced separately and combined by creating, after the production of that A chain and B chain, they combined it with disulfide links. That we know that by combining disulfide links, that will become insulin. So that was the plan made by a Lilly company, that we now we are producing insulin hormone. So Escherichia coli, the two gene sequences, different DNA sequences, coding for A gene, coding for that B polypeptide, B gene, A gene. In that way, we can talk about. They produce it separately and they attach it by disulfide links. That produced that make the production of insulin, and that insulin we produced it here, the humulin. That insulin we produced here, the humulin. You might have learned over the recombinant DNA technology with PBR322 plasmid, that vectors, casmate, restriction endonuclease, enzyme participation, ligase as attachment, that all the things you might have learned over in molecular basis. That's why I'm not at all focusing that area. Just I am showing that how by administering the recombinant DNA technology, how we produced the insulin, that only in that area we just focused over. As long as this technology is going to be improvised over, the technological improvement of this recombinant DNA technology, you might have studied over, it is depending upon the number of restriction endonuclease enzymes which we are going to identify. The restriction endonuclease enzyme discovery is going on, and already you might learned over 200 plus restriction endonuclease enzymes are that are already recognized, and it's going on. As long as this go on, that we can cut a specific DNA at a specific place, and we can use more number of products like this, which are useful for the human being in the future days. That is biotechnology. Biotechnology, we can say that the applications of the biotechnology already now it is used. Yes, this topic is focusing only on the insulin insulin structure in a general way, and what kind of problems we face for the production of insulin, and then how does that Lilly company has solved that particular problem, how does that particular insulin is available nowadays in that market. So that application question from here, that why we can't take the insulin in oral way, in through for that through as a capsule form, why we can't take that? When we ask, we know that it's a protein hormone. If you remember it as a protein hormone, that will be which you can easily say. So that's it about the biotechnology applications. Insulin synthesis, we said, we learned.